Good morning. This is Ann Buckland. I'm chair of MetaZooGene SCOR Working Group 157, and the leadership team also includes two vice chairs, Katja Pijnenberg from the Naturalis Biodiversity Center in the Netherlands, and Ksenia Kosipakova from the Shurtsov Institute of Oceanology in Russia. MetaZooGene now includes 23 members from 19 countries. We have excellent global coverage, which is a real advantage of SCOR, of course. Our terms of reference include an open access web portal for DNA barcodes, optimal DNA barcoding pipeline, best practices for DNA metabarcoding. All of this is focused on marine zooplankton biodiversity. Our deliverables include a project website, web portal, best practices, publications, review papers. Our capacity building activities have primarily focused on inviting early career colleagues to all of our activities, meetings, conferences, including a symposium that we sponsored in association with the 2022 ICES Annual Science Conference. Deliverable number one, term of reference number one, is an atlas and database. This was created by Todd O'Brien, a MetaZooGene member from NOAA Fisheries in the USA. We started with a database of cytochrome oxidase 1 sequences, barcode sequences, focused on marine invertebrates. And our current record is about 30% coverage of the more than 66,000 species that are recognized by Todd's Copepod Obus website. Todd has expanded it now for additional genes used for barcoding and identification of taxa in the pelagic, for metabarcoding microbes to mammals now. And you can see here examples of coverage, both for taxonomic coverage and for geographic coverage. In 2024, Todd led a publication in a invited chapter of a Springer Protocol book, which basically allows readers to provide detailed instructions and guidance for users of the MetaZooGene Atlas and database. It's a useful resource to make sure that there are alternate sources in addition to the website. In 2021, we published a summary of the CO1 barcode status of marine zooplankton. This was a group effort by all of the MetaZooGene members, and it is a successful update. It's been cited more than 100 times since 2021, and we think it's a useful contribution from the working group that basically did take advantage of all of the expertise we have among the 23 members. Publications, I mentioned, are one of our primary deliverables, and this is because, of course, the SCOR members are typically mid-career academics, and publication is essential for their own success. We focus on this. To date, MetaZooGene members are listed as co-authors of 51 peer-reviewed publications from 2019 to 2024. All of these, or most of these, do informally acknowledge the collaboration with MetaZooGene, but the SCOR formal acknowledgement of funding is cited for 14. Early career authors are included as co-authors on 44 of the 51, and this is just an update with some of the more recent papers that have been published. Ongoing now to meet deliverable 3 is what we call an intercalibration experiment. What that means is basically 10 MetaZooGene Working Group 157 teams extracted DNA from zooplankton samples of their choice. We exchanged those 10 samples among the 10 labs, and each of the participating labs sequenced the 10 samples for 4 genes, and then submitted the data to a central depository, and that the bioinformatics statistics has been carried out with a lead by Leocadio Moncobercial at ASU, And the whole goal of this is to evaluate the reliability and accuracy of DNA metabarcoding for analysis of zooplankton diversity throughout the global ocean, because of course metabarcoding is being used widely now for fisheries management and assessment, and it's important that we understand exactly how to make it as reliable as possible. This is a peer review publication that we're preparing for this year. MetaZooGene was also named as an Ocean Decade Action, and our project is assigned to the Marine Life 2030 program in the Ocean Decade, and that coordination is led by Leocadio Ponca-Rusial. We are also affiliated with two other programs, the Decade Collaborative Center in the Northeast Pacific, DCC-NEP, that coordination led by Jen Questel, and with the Ocean Biomolecular Observing Network, OBON, and that coordination is led by me and Penny Lindequay from Plymouth Marine Labs. In 2024, we held what will be our last annual meeting. We asked for a fifth year for SCOR sponsorship to allow us to finish up our deliverables. This was a high point, I must say. This is 40 people attending the working group meeting in association with the Pisces Zooplankton Production Symposium in Hobart, Australia. It was hosted by the University of Tasmania. We had guests from that home institution, in addition to the 14 working group members and 10 early career colleagues, as we call them. It was really a high point to allow us an open discussion on future goals, how we're going to keep moving when Working Group 157 finishes this year. With that, I'd like to acknowledge all of the MetaZooGene Working Group 157 members, the support from SCOR, which has been invaluable, UN Ocean Decade Science, and also the International Council for the Exploration of the Sea, ICES, which is a partner institution for us, as well as NOAA Copepod and others. Thank you very much for your attention.